level is an example of negative feedback that do not utilize the hypothalamic pituitary axis. For example, insulin regulates blood glucose concentration. In turn, insulin secretion is turned on or off by changes in blood glucose concentration. Here, when there is high glucose concentration in blood, beta cells of islets of Langerhans secretes insulin which act on liver and lower the blood glucose concentration. Insulin has following actions: increase glucose uptake among several target tissues, increases cellular respiration, increases glycogen formation, increases triglyceride synthesis in adipose connective tissue. But when there is low blood glucose concentration, alpha cells of islets of Langerhans secretes glucagon which acts more, that is positive control, on liver and increases blood glucose concentrations. Glucagon has following actions: increase glycogen production in liver, increases glycogen breakdown in liver and muscle, increases triglyceride breakdown in adipose connective tissue. In this way, blood glucose concentration is maintained. Water balance and antidiuretic hormone, that is ADH. ADH or vasopressin plays an important role in the body's water and electrolyte economy. Thus, ADH release promotes the reabsorption of water from the urine in the kidneys. Through this mechanism, the body reduces urine volume and conserves water. The main stimulus to ADH release into the pituitary gland is an increase in plasma osmolality, which is detected by osmoreceptors, that is water receptors in the hypothalamus. ADH release from posterior pituitary acts on kidney. In response to elevated level of ADH, the distal convoluted tubule and collecting ducts synthesize proteins that make the tubules permeable to water. As water flows through the tubules embedded within the salty medulla, water is reabsorbed by osmosis. The other important stimulus to ADH release is a reduction in the effective circulating volume, that is hemorrhage. This is detected by baroreceptors, that is pressure receptors in the aortic arch and carotid sinus. Once plasma osmolality is returned to normal, a negative feedback mechanism inhibits further ADH release. Only a few positive feedback loops occurs in the human endocrine system. One such example is the process of milk release from the mammary glands. Hormonal methods. Hormones, usually a peptide, amino acid derived or steroid, produced by glands and conveyed by bloodstream to effect physiological activity such as growth or metabolism. Many hormones can be measured, that is acid, or identified in the blood to give an indication of metabolic processes and conditions or hormone imbalance. These include male and the female sex hormones, that is estrogen, progesterone, testosterone, and hormones secreted by other glands such as pituitary, parathyroid and adrenal glands. There are several types of hormonal assays. Some common assays are radioimmunoassay, that is RIA, enzyme immunoassay, that is EIA, or enzyme linked immunoadsorbent assay, that is ELISA, that is ELISA, radioreceptor assay, that is RRA, immunofluorescent assay, that is IF, and immunohistochemistry. Radioimmunoassay. Radioimmunoassay allows for the measurement of wide range of materials of clinical and biological importance. This technique has a significant impact on medical diagnosis due to the ease with which the test can be carried out while assuring precision, specificity and sensitivity. The radioimmunoassay technique, as the name implies, achieves sensitivity through the use of radionuclides and specificity that is uniquely associated with immunochemical reactions. Yellow and Burson were awarded the Nobel Prize for pioneering radioimmunoassay when they evolved radioimmunological assay for insulin. At about the same time, Roger Kane also published his work on determination of serum thyroxine using a competitive binding assay. In later system, the naturally occurring thyroxine binding globulin, that is TBG, was used as binding protein, and radioactive label thyroxine, that is T4, was used as tracer. Through not strictly an immunological test, the principle of competitive binding of compound under test to a specific protein remains the same. The radioimmunological determination of thyroid stimulating hormone, that is TSH, was demonstrated by Odell and thyroxine by Murphy. In principle, a known quantity of an antigen is made radioactive frequently by labeling it with gamma radioactive isotopes of iodine such as 125I attached to tyrosine. This radio-labeled antigen is then mixed with a known amount of antibody for that antigen, and as a result the two specifically bind to one another. Then a sample of serum from a patient containing an unknown quantity of that same antigen is added. This causes the unlabeled or cold antigen from the serum to compete with the radiolabeled antigen, that is hot, for antibody binding sites. As the concentration of cold antigen is increases, more of it binds to the antibody, displacing the radiolabeled variant and reducing the ratio of antibody radiolabeled antigen to free radiolabeled antigen. The bound antigens are then separated from the unbound ones, and the radioactivity of the free antigen remaining in the supernatant is measured using a gamma counter. Using known standards, a standard curve can then be generated which allows the amount of antigen in the patient's serum to be derived. ELISA, enzyme linked immunosorbent assay, that is ELISA, are plate based assay designed for detecting and quantifying substances such as peptides, proteins, antibodies and hormones. Other names such as enzyme immunoassay, that is EIA, are also used to describe the same technology. In an ELISA, an antigen must be immobilized to a solid surface and then complexed with an antibody that is linked to an enzyme. Detection is accomplished by assessing the conjugated enzyme activity via incubation with a substrate to produce a measurable product. The most crucial element of the detection strategy is a highly specific antibody-antigen interaction. ELISA can be performed with a number of modifications to the basic procedure. These are direct ELISA, indirect ELISA, sandwich ELISA, and competition or inhibition ELISA. Radioreceptor assay. Significant advances in understanding the actions of hormones, neurotransmitters, and drugs resulted from the development of new analytic procedures. Among these advances, the discovery that for a given drug or hormone or neurotransmitter, there exists receptor site on membranes to which the substance must attach in order to evoke a biological response led to the setting up of the radioreceptor assay. Thus, ligand binding techniques can be used as assay for measuring drug levels in biological tissues and fluids. Radioreceptor assays are based on the principle of competitive protein binding methods. When a radioligand and an unlabeled ligand are present together with a specific receptor preparation, the amount of radioactive ligand bound to the receptor is a quantitative function of the amount of unlabeled ligand present in the incubation medium. The technology of the radioreceptor assay is simple and rapid to perform when optimal conditions for binding are determined. Radioreceptor assays are highly sensitive, reliable, precise, and accurate. The chief limitation relates to specificity, as any substance having an appreciable affinity for the receptor displaces the specifically bound radioligand. Paradoxically, this lack of specificity may be in some cases advantageous in that it allows for the detection of the parent compound and active metabolites in proportion to their affinity for the specific receptor. During recent years, radioreceptor assays have been applied to quantitative determination of hormones, neurotransmitters, and drugs. Immunofluorescence. Immunofluorescence is an antigen-antibody reaction where the antibodies, that is monoclonal, are tagged, that is labeled, with a fluorescent dye, and the antigen-antibody complex is visualized using ultraviolet, that is fluorescent microscope. Fluorochromes are dyes that absorb ultraviolet rays and emit visible lights. This process is called fluorescence. Commonly used fluorochromes are acridine orange, rhodamine, etc. However, these fluorochromes are used for general fluorescence. When fluorescein FITC is excited by a blue light, that is wavelength 488 nanometer, it will emit a green color, that is at wavelength 520 nanometer. Phycoerythrin PE emits an orange color at 570 nanometer wavelength. The fluorochromes commonly used in immunofluorescence are fluorescein isothiocyanate, that is FITC, and the tetramethyl rhodamine isothiocyanate, that is red. Immunohistochemistry. Monoclonal antibodies are tagged with fluorescent dyes and are reacted with the cells or tissue pieces. After washing, are subjected to fluorescent microscope to identify specific peptide or protein present in the cells or tissue pieces of interest. Let us summarize. Endocrine glands are able to synthesize, accumulate, and release of special hormones into circulation at optimal levels for their physiological actions in the target tissue. But their optimal levels are regulated by hormones, nerves, and metabolites by feedback mechanisms for homeostasis. Such hormonal molecules are measured and identified by techniques like RIA, ELISA, RRA, immunofluorescence, and immunochemistry at micro levels. Thank you.